Okay, do we have any questions before we get started? Any questions at all? Course mechanics, those of you that just came in, please take the Socrative exam. There's your quiz right on top. Okay. So let's do a quick review from last class. We're talking about translation. How do we know when you have a messenger RNA like the one you were just working on or the one that you're working on right now, how do you know where translation starts? If it's a eukaryote, it's a start codon. What's the start codon? AUG. Okay. Which AUG? There could be a lot in a messenger RNA. Say it a little bit louder. The first one to the right of the 5' cap. That's what I heard up here in the front. So the first AUG you encounter reading from the 5' end. That's eukaryotes. How do we know where translation is supposed to stop? I know it's the question that's got the obvious answer, so no one's saying. What is it? Stop codon. But it's not just any stop codon.
It's got to be a stop codon that fits the reading frame.
It's got to be a stop codon that's in the reading frame, right? The first AUG tells us which sets of 3 nucleotides make up each codon. So you don't just read left to right looking for, oh goodness, what are the 3 stop codons? UAG, UAG, and UAA? Oh, I said this. I wrote the same thing twice. How bizarre. UGA and UAA, is that right? Okay. So when we know the start codon and the stop codon, then we can tell how many amino acids are encoded, how many codons there are. So that's the position of the start codon and the stop codon. Then we know how many amino acids there will be in this protein. Okay. Last class, this was one of the 2 quizzes that we did. I'm showing you in blue numbers there how many of you responded to each of these possible answers. So we had a 15 nucleotide CDS coding sequence here. And I wanted you to predict what would happen if we had an insertion mutation, so an A and a G happen right there between nucleotides 4 and 5. There's a variety of answers. Nobody picked, why did nobody pick D? Poor D. What is it about D that you don't like for an answer to this question? What happens to the protein that's encoded by this messenger RNA with this mutation? So why not choose D? Pardon? So if you're adding nucleotides, then why not D? The length has to change? Is that true? Depends. So every time you're faced with this sort of question, I'm not saying that D is the right answer. But every time you look at one of these questions, and that's the exercise that we're doing in class today, is to see how mutations affect the lengths and the amino acids encoded by proteins. So let's actually translate this really quickly. So if this is this eukaryotic? Did I say? I didn't. Let's assume it's eukaryotic. It's a coding sequence. So what's the first codon going to be? By definition, a CDS starts with methionine, the start codon. Okay, so that's M. After this insertion happens, what's GAG? Which amino acid is GAG? So which one? Glutamic acid, E. Hey, we've got me. Okay. So GAG, that G, those two that are inserted, what happens next? What's the next set of 3 nucleotides? UA, yeah, so in a coding sequence, we'll get to this in just a second. UAG or TAG then are the next 3 nucleotides. That's one of the stop codons. So after this mutation, there's an M and then there's an E, methionine, glutamic acid, and then a stop codon. So we get a protein with 2 nucleotides. So is that shorter or longer than the original protein? It's shorter. The original protein with the CDS always starts with the AUG, the start codon. What does it end with? Which codon? The CDS sequence always starts with the start codon and stops with the stop codon. So the original messenger RNA, the CDS, the coding sequence, had 1, 2, 3, 4 amino acids. So this mutation, despite making the mRNA sequence or the CDS sequence longer, actually produces a shorter protein. So that makes answer A the right answer in this case. The protein with this mutation becomes shorter than normal, and its amino acid sequence does change. Any questions about this? So anytime you're faced with a question, don't just go with your gut instinct about whether or not the encoded protein might be shorter or longer, change its amino acid sequence or not, translate it and see what happens. That's the bottom line. The other example we encountered last class was trying to just simply figure out, and simply, every time you see a question that looks simple, it's probably not, arrange these 4 CDSs in order of how long the proteins are. In this case, it was actually the expected answer. Because these are CDSs, they all start with the start codon. They all stop with the stop codon. So in this case, you really can just look at how long the sequences are, and that's directly related to how long the proteins that they encode are. So this is the longest, the second longest, the third longest, the fourth longest. But I loved the fact that a lot of you caught me on this point. To those of you that wrote something to the effect of, wait a second, none of these have AUG, the start codon, I got something to say. You're right. CDSs are written in DNA nucleotides. So the coding sequence of a gene is written in DNA. So every time you see a T, you should just think U when this is transcribed into RNA. So you're right. Technically, there was no AUG, there was no start codon, but in the CDS format, the ATG is the AUG. Concerns or questions up to this point? Okay, so this should look really familiar. What's the answer? Is it A or B? Let's check. Oh dear. Well, let's see. If you can stare in between the gray lines, I'll tell you the results. 60% of you said A, and 40% of you said B. So who's going to be right in this case? Here's the practice for translation. What do you have to do to translate these messenger RNAs? What's the first thing you do? Find the start codon. These are eukaryotic, so we read left to right. Where's the start codon in A?
After the 2 nucleotides.
Okay, so after the first 2 nucleotides, there's an ATG. That's methionine, always. What's GCA? Alanine, Ma. Then what?
Stop codon.
Tag. Stop codon. So the first protein produces Ma. The first messenger RNA produces Ma. What about the second, number B, letter B? First ATG from the left, the 5' end, M, and then a stop codon, TGA. So A produces the longest protein. Okay. That's it for the review, right? Just simple, here's a messenger RNA sequence, translate it. That gets us through most of the outcomes for this section, this chapter on translation. We've translated messenger RNAs, we've predicted effects of mutations on protein sequence. Today, hopefully everybody brought your mRNA sequences to class. We'll work on them in class maybe a little bit. They're useful for over the weekend as well. We're going to predict the effects of mutations that you make to your CDS that you brought in for class today. First, I want to make this more applicable, perhaps, in terms of why it's important to know something about how translation works to be able to see what sort of effects it has on organisms. For example, let's take maize, corn. The corn that we have today was originally, thousands of years ago, a wild plant called teosinte that looks like this. Which one would you rather try to eat, teosinte or maize? I don't know. Depends on whether or not you've got a big mouth, I suppose. There's an important difference between teosinte and maize, and this was seized upon by ancestors of ours and yours who were domesticating maize. So this is teosinte on the left, this gentleman standing next to this plant. That's what a teosinte plant in all its glory looks like. These little tiny little things that kind of look like corn cobs, but they're basically no good to eat. And all of this domestication, maize, or teosinte being domesticated and becoming maize, originated in southern Mexico, a little bit tropical. And some scientists went and looked underneath this humongous boulder, right? So here's a boulder, there's a gap, there's an empty space underneath the boulder that was originally a shelter for people that lived there about 10,000 years ago. They did some excavation underneath this ginormous rock, which I don't know if I'd crawl under there and do any excavating. I suppose if it's been like that for 10,000 years, it might be safe. They found these milling tools that people had been using to grind maize, in this case, to make flour. And something I didn't know they could do, they actually were able to extract plant material from these stone tools and look at it under microscopes and do analysis of it, and they could determine from the starch granules and the shape of the starch granules and some other aspects of the plant material they found that at this point in time, these stone tools were dated to about 8,700 years ago. They had maize-like, corn-like properties. So that suggested that by 8,700 years ago, ancestors had domesticated what was teosinte into maize. So this is an important point because what we're doing here is we're going to look at how genotype tells us something about phenotype. This is a really cool example. So again, we've got teosinte panel B here on the left side. It has these closed fruit cups. So the fruit itself, the corn, is totally enclosed by a really hard capsule. With a single point mutation, one nucleotide change, scientists can move that one nucleotide change that maize has that teosinte doesn't into teosinte, and this is the effect. So you make one mutation, you go from B to C. A single nucleotide change makes plant B look like plant C. And the difference there is that plant C, as it says there, has open fruit cups. So it's much easier now for people to get access to the fruit. It's not covered in this really hard shell. It's exposed. Makes it easier to eat. Don't worry about seeing what's projected. DNA sequence analysis. So what these individuals were doing was they took a bunch of individual maize plants that I'm circling in red and some teosinte plants, current teosinte plants. So we're comparing corn to its ancestor. And they wanted to know, is there some specific DNA sequence difference between maize, corn, on the top in red, and teosinte, its ancestor? Is there some mutation that can explain how all of a sudden we went from this tiny little plant where the fruits are all covered up to corn where the fruits are a lot larger and they're exposed and they're easier to get access to for us? So on your tablets, maybe it's easier to see if you zoom in. Look across the very top here. These numbers. What do the numbers refer to, do you think? There's a DNA sequence here, and then we've got numbers. How many of the numbers are positive numbers? 1, the one on the far right, it's number 18. Okay. So if that's nucleotide 18, is it being transcribed or not? It's plus 18. So yeah. All the rest of these are negative numbers. Where are those negative numbered DNA sequences located in the gene? Which part of the gene? You start numbering at +1, that's the first nucleotide that gets transcribed. Everything to the left, minus numbers, must be in the what part of the gene? It's untranscribed. So what's the left of the transcribed part of a gene? Starts with P. Yeah, say it louder. The promoter. So all of these DNA sequences they're looking at, all the negatively numbered ones, they're looking for DNA sequence changes in the promoter of this gene. The gene called TGA1. This is the gene that controls whether or not the fruits are closed or open. There was only one place in all of this DNA sequence they analyzed where there was a perfect correlation between the DNA sequence that you have if you're maize and the DNA sequence you have if you're teosinte, and that is position 18. Every time you see a period, that doesn't mean that that nucleotide is missing. That just means that it's the same as the one above it. So that means every single maize plant they looked at, the phenotype being open fruit, every single individual had a C at that spot in this gene. But when they switched to a different plant, they looked at teosinte. Which nucleotide did every teosinte plant have? They have a G. So there's a perfect correlation between what DNA nucleotide you have at this one spot in this one gene and whether or not you've got open or closed fruit, accessible or not. Now remember when we talked about correlation versus causation? Per capita consumption of cheese is perfectly correlated with death by strangulation in bed sheets. That graph that we looked at a couple weeks ago. So this is just correlation. This doesn't prove that this DNA sequence causes this change, but there's a perfect correlation between whether you've got a C or a G at this spot, nucleotide 18. See what it does. So in the abstract of this paper, the authors say that here, and we saw this in the previous table, at position 18 in the transcript, there's a G in teosinte, there's a C in maize. The authors also tell us in the abstract that the amino acid that's produced when you have a G is K, that's lysine. And if you have a C at position 18, that makes amino acid N, asparagine, or ASN. So your job right now is to tell me if teosinte has lysine, which of these nucleotide positions that are blank here can you fill in? 16, 17, 19, or 20? So how would we do this? We're looking for a lysine codon. So where are the lysine codons in the codon table? Let's pick those out first. Are there any other ones other than those two? Nope, just those two. So it's either AAA or AAG that's making lysine in teosinte. Which one is it? It's got to be AAG, because it's a G we're working with. So this author's told us there's a G that's part of making lysine. There's only one codon choice for lysine that contains a G. It's AAG. So does that make sense, given the fact that these authors have said that if you change a G from a C, you make asparagine? Is that true? If you go from AAG to AAC, where's AAC? Yep, right above it. Right. So a single nucleotide change causes an amino acid change because it's in the third position of the codon. Circled here in red. If you start with AA, there's only two types of amino acids you can make, lysine or asparagine. And this simple change, last point here, that simple change, one amino acid change caused by one nucleotide change causes teosinte, K here in this panel, lysine. You make an asparagine. That one change makes this big of a difference in how the fruit of this plant looks. Panel B to panel C.
Is that because the different amino acids changes the conformation of the protein or how it folds that produces those types of shells?
So that's the fabulous question to ask next. So why would a single amino acid change make that kind of a difference? Does it cause, as was asked, does it cause the protein to change its structure and that affects its function? What does this change do? Don't know yet. But that's the perfect question to ask next. Have you taken biochem yet? Oh, I was going to say, ask your biochemistry teacher. That'll stump them. Like, okay, I got to go read this paper first and find out what they're doing. So in this paper where they published this, they didn't yet get to the biochemistry of it. It's a great question. So you can predict how a mutation at the nucleotide level can affect a protein. What we can't do then is predict how a protein might affect the function of an organism. We've seen one other example of this. This I just wanted to point out because I love the earwax example. You saw this on one of the tests. You might not remember it because you might have test amnesia. Right? This was another example where there's a single nucleotide change in humans that dictates whether or not you've got wet earwax or dry earwax. So another example of how a really, really tiny single nucleotide change can have a visible effect on phenotype. So this is why I'm so interested in having you understand how to take a messenger RNA sequence and translate it and understand what's the protein, how is the protein affected by a mutation. Any questions or concerns? Okay. So hopefully this is all true. You found a gene, you brought it to class, and you've got the text version of that CDS, the coding sequence, starting with the start codon, ending with the stop codon, somehow in a text format, so you can copy and paste it into other documents. I'm going to walk through pretty briefly, like we did last class, the next step in this workflow to analyze this messenger RNA CDS sequence with you. So watch this. Again, as always, I'm recording this. It'll be on YouTube, so if you want to watch back through what analysis, how to do this analysis, and what to do next, you can do that. This is the CDS that I have, the NUO1 gene. I don't know what it does. It's just a gene I found. It starts with, as we've already seen multiple times today, it starts with the start codon, it stops with the stop codon. What I want to know is, what's the amino acid sequence? And that's what I'd like you to be able to do first. So this is how I'm going to show you how to do that. Use this website, You can take a picture of the QR code if you want, or just click on the link. If you've got the link, type it into a browser later if you like. I warned these people that we were coming, so they're expecting 65 students to log into the website and be hammering their server with this analysis. But it's pretty straightforward. It's a simple analysis. It's just a computer program that was written that takes a messenger RNA sequence, uses the codon table, and spits out the protein sequence. But I don't want you to spend your time doing that necessarily, especially on an exam. So here's a website that does it for you. When I ask, here's a 1,000 nucleotide messenger RNA sequence, please translate it. Don't do it by hand. I never would. So why should you? So use this tool. Here's how to use it. When you go to this website, it's simple. Box number 1, paste in your CDS. That's it. Paste in the whole nucleotide sequence from ATG to the stop codon into box number 1, the red arrow. Okay. Hit submit down at the bottom. That's all you have to do. Computer chugs away for half a minute. So there's me filling it in. I pasted in my sequence, I hit submit, and it gives me the amino acid sequence encoded by that CDS. So you can work on this now, you can work on it later. It doesn't have to be done now in class. When you get that translation, when the computer program, when the website spits out your amino acid sequence, copy that text to a text file as well, because we're going to do a little manipulation. Okay. Not right now, but when you're there. Copy and paste the text of the amino acid sequence. The goal is to figure out how many amino acids are in the protein. That's the first step. I want to be able to have everybody do that as well. And again, I don't know, what do you think? What's the easiest way to count the number of characters in a text file? Use a word, right, so that's what I do too. I paste it into Word, I use the character count command, except I don't have Word. Now, if you want to do any of this, by the way, on laptops, home computers, desktop computers, that's totally fine. You don't have to use a tablet to do this. Use whatever computer you want. If you're on a tablet, though, I don't have Word. Google Docs on a web browser has a character count command, but Google, the app, doesn't. Go figure. So access a simple website, charactercounter.com, same thing. There's a field. You paste in your text, it tells you how many letters are in it, right? So when you want to know how many nucleotides are in a nucleotide sequence or how many amino acids are in a protein sequence, I'm not at all financially affiliated with charactercounter.com. You could use whichever, there are lots of these websites that do this. It's just the first one I found. So once you translate your amino acid, your protein, as I did, I pasted it in, and down here at the bottom, I want the without white space number. We don't want to count spaces. So there are 479 amino acids in this sequence. So NUO1, the gene, encodes a protein that's that many amino acids. It'll become clear pretty soon why I'm so interested in how many amino acids are in a protein. Any questions? So take the CDS sequence, use transseq to translate it into amino acid sequence, and then you can count how many amino acids are in that sequence. Pause for a second, if you can. This is where it gets way better. This is better than maize by far. I'm trying not to be corny. Oh, don't get me started on, sorry, I already got myself started. There are so many puns you can make with corn, it's not even funny. I've got an ear for puns. Oh. So what I'd like you to do is you brought your CDS sequence with you to class. I want you to mutate it. So I'd like you to have the original version, the original sequence of that CDS in a text file. Make a copy of it, so you've got two versions. And I'd like you to make one mutation to that gene you brought with you, one type of mutation. So copy paste that text of the CDS, second copy, and then follow the rules. So choose to do one of two things. Either insert some nucleotides into that CDS or delete some. One or the other. You pick. And then if you're inserting, insert some nucleotide sequence you make up. 1 to 12 nucleotides in length. Doesn't matter. 1, 2, 3, 4, whatever. You pick. Write out that many random nucleotides or non-random nucleotides if you want. And stick that into the CDS somewhere. You decide where. If you want to delete some nucleotides, do exactly the same thing. Pick 1 to 12 existing nucleotides in the CDS, cut them out. So please work on that now if you can. We've got time for this. So take 3 or 4 minutes. Raise your hand if you've got a question. I'll come around and answer technical questions. And work on this mutation just for a minute. 2 or 3. I'll come around and answer questions. CDS, DNA sequence. Yeah, which is fine. So the translation website will translate T's instead of U's, just fine.
Okay.
So you don't have to worry about that. Yeah. For the next step, you need to know your code down here at the bottom. This is why it's important for you to have decided how many nucleotides you're going to mess with and whether or not you're inserting or deleting. So remember, if you're inserting, you're an I. Everyone gets a designator. If you're deleting, you're a D. And then if you're doing 1, 4, or 7 nucleotides, that makes you a 1. If you're deleting or inserting 2, 5, 8, or 11 nucleotides, you're a 2, and so forth. So everybody figure out which code you are. We're going to get into groups. So I'm going to ask I1s, D1s to go in different parts of the room. So figure out which of these you're going to be. Is everybody okay with that part so far? Any questions about your group? Okay. Lastly, I need you to make predictions. So this is all about understanding how mutation affects the protein you're working with. So given whether you're making insertion or deletion and the number of nucleotides you've decided to modify, make predictions about these 2 questions. And then I'm going to open the next Socrative quiz. You can put the answers to the 2 predictions and tell me your group code. So by yourself, make a prediction, 1 and 2, and then take the Socrative quiz. Okay, let's come back together briefly. It sounded like every group, or some of you split up into smaller groups within your groups, which is fine. Let's wrap up just quickly. So I want to have everybody familiar with what all of the predictions from all of the groups were. So, group I1, who wants to represent insertions of 1 nucleotide? Some brave soul. Did you predict that your protein was going to increase, stay the same, or decrease?
It's going to increase.
Okay.
Because when you insert a nucleotide, you could probably end up having a longer protein because you added a nucleotide sequence, so you could probably add an amino acid to the protein chain.
So this is a group that added 1 or 4 or 7 or 10 or that's it, right? 1, 4, 7, or 10 nucleotides. So that addition might make the protein increase in size. Does that change the protein sequence?
Yes.
Yes. You don't have to use my weird arbitrary severely or barely. We'll just say yes. It's predicted to change the sequence of the protein. So what about I2? Same question. Is it going to change the size of the protein or not? Decrease, increase? Did anybody insert 2, 5, 8, 11 nucleotides? Really, out of a class of 75 students. What's the probability? Okay, now we have to do a chi-square test. We'll skip I2 for just a second. I3. Some representative of I3. What do you think? You insert nucleotides, multiples of 3. What did you think? Yeah, you split up into different groups. Anybody from I3? Don't make me call on somebody.
You could put a stop code on, like, in between, and then it could shorten the sequence of the loss.
You might.
Put it, like, towards the end, and then it would make it longer, and it wouldn't be severe. It would, like, barely change it.
So it's complicated.
Yeah.
It's complicated. Okay. How about D1s? What's the prediction? You delete 1 nucleotide or 4 nucleotide or 7 or 10, what does that do to your protein? You think? Shortens it? Okay. Does it change the protein sequence? Probably. So we're going to say yes. What about D2s? The same? So if you delete 2 or 5 or 8 or 11 nucleotides.
We're the same as them. Yeah.
Oh, you're the same.
No, like.
Just the same answers.
Yes.
You're clearly not the same. I know you're genetically different. Oh, let's see. Sorry. Okay. What about you folks back here? What would you like to tell me about what happens when you delete multiples of 3 nucleotides?
You're going to decrease your protein length
You decrease the protein length.
and also change your protein sequence, but not a lot.
Okay.
Because it's a multiple of 3.
So we'll say yes, it changes the amino acid sequence, but barely. Is a pattern emerging in the predictions? Where's the strongest pattern? If you delete, everybody's predicting that if you delete, it decreases the protein length and it changes the amino acid sequence. If group I2 were here, let's pretend we all inserted 2 nucleotides. Let's all act as group I2 without the talking. Is inserting nucleotides going to change this protein sequence? Okay, I'm hearing a few people say yes. And we're just going to assume that it will also change the protein sequence. Maybe severe, maybe barely. So here's a prediction. That if you made one of these types of mutations, now you all get to benefit from everybody else's thinking. This is the prediction. Now is when I want you to, over the weekend, take that mutant CDS you just made by inserting or deleting nucleotides, run it through the translation website, count the number of amino acids that are in your new protein, and compare that to your prediction. For the random mutation you made to your gene, did it actually meet your expectation?
Dr. Ross?
Yep.
For any of the mutations that we're talking about,
Yeah.
do you have to take into consideration the number of sequences in there? Let's say you have an odd number of nucleotides and you make one insertion, then you end up, that insertion might not end up making a difference because you're going to have, it's still an odd number, but having, if you have not a number but you answer one, you're going to end up having an even number, so now you might not end up doing anything.
So you do it. So the number of nucleotides does matter, and that's why when we come back together on Monday, we're going to compare. That's why this is important for doing by Monday. Bring back your information. We're going to compile everybody's observations from your mutant genes. We're going to see what happens. One other point, hang on. One other point in blue at the bottom of the slide. We've got about a week to go until the next exam. So if you would like to contribute questions for the review sheet, please do so by Monday night, okay? It's a week from Monday, the exam. Okay. Thanks, everybody. Have a good weekend.